Welcome to MOOC course on Introduction to Proteogenomics. It is well known fact by now that most diseases are caused due to dysregulation of pathways or networks, and they are not caused just because of effect of a single gene. It could be just in very few rare cases, but otherwise it is a group of genes or proteins or a pathway is actually going to affect a given disease. Hence it is important to understand how proteins interact with each other. In today's lecture, Dr. Bing Zhang will introduce you to the concept of network analysis and various tools available for its use. I hope this information will be very helpful for your own projects when you are looking at how proteins interact with other protein and form a given network. So, let us welcome Dr. Bing Zhang for today's lecture.
So, in this lecture we are going to talk about biological network analysis. So, I think we had plenty of science and formulas and things like that. So, I will start this lecture with a poem. Actually, it was a very beautiful poem from poet John Donne, and this was written 400 years ago. It's titled "No Man is an Island." And I don't want to repeat this, but it's a very beautiful piece of work and basically express the idea that the holistic view of the society, so no man can survive or thrive without the support from the community. So I think it's still true, although 400 years has passed, it's still true in today's society. And you can think about people around you, for example, through connections with WhatsApp. I think that's the one you guys use in India. You have a connection through that social network, right? And so a few years ago, when I was reading a review article in this Nature Review Genetics from the Albert Barbasi, so basically this sentence reminded me about this poem. And the idea is that if there is a change in a gene in the network, it will have the impact not only to the gene itself, but the impact will pass through the whole network. So I was thinking, okay, so it's not only no man is an island, actually no gene is an island. So that's the thing we're going to talk about today. So we need to understand biology, understand the genes in the context of the networks. So indeed, during the past decade also, a lot of studies have shown that complex phenotypes, including most of the disease phenotypes, are the result of the dysregulation of the networks rather than individual genes. Like network dysregulation causes disease, not individual gene cause disease. And in order to understand the network, I will first introduce two terms. One is called node and the vertex or vertex. So those are basically the individual elements in the network, and they are connected by edges. So with these two terms, then we can look at some typical biological networks we have. They can be divided into two categories: the physical interaction networks. So these are the types of networks, the nodes are connected to genes or proteins that physically interact with each other. For example, the protein-protein interaction network. And in this network, each node is a protein, and the edge represents the interaction between two proteins. And the signaling networks, and it's a specific type of protein-protein interaction network. And it's not only the interaction of the proteins, but the protein A can kind of regulate protein B, right? For example, a kinase, as we just talked about, kinase could regulate the downstream target. So in this way, you can imagine a network like this, but the edges are directed. For example, this is a kinase, this is a target. This is called undirected protein-protein interaction network. But in signaling networks, it's this protein can modify the other proteins, so there is a direction associated with the edge. And also the gene regulatory networks. In these networks, the nodes are either the transcription factors, or it can also be the microRNAs, and they'll target genes it's also a directed network. So it represents the physical interaction between the TFs or microRNAs and their targets. And the metabolic networks, in this one, the loads are the metabolites and the edges are the reaction going from the substrate to the product. Also, it's a directed network. Another type of network is called functional association networks. In this type of network, we don't really know whether the two genes in the network interact with each other or not. They may or may not interact with each other. For example, the co-expression network, if we have a lot of experiments, and we always see two genes keep going up and down together, we can infer the co-expression relationship between them, and that can actually be quantified, for example, based on the Pearson correlation. And in this way, we can also call this a weighted network, meaning the edges can be weighted by the co-expression level, right? And it's also undirected. And the genetic network, those are the genes, and the relationship indicates the genetic interaction, meaning you do some perturbation experiment. If you knock out gene A or gene B, you get the same phenotype, then you can guess maybe there's some relationship between the genes. So the first question I want to talk about is how we get all those networks. Because we talk about the network, we need to get the network first in order to do something on them, right? So first we talk about the protein-protein interaction network. And in order to get protein-protein interaction networks, basically we want to establish the relationships or interaction relationship between two proteins, as you can see here, right? So there are experimental approaches that can help us to get this type of relationships, including the Is2 hybrid experiment or the pull-down experiment, pull-down followed by the mass spec analysis. And there are also computational approaches that can help us infer the protein-protein interaction relationships. For example, we can start from the known protein-protein interactions, and then we can try to infer, which domains actually interact with each other, and then we can generalize to new protein pairs. If they have those interacting domains, we can guess maybe they interact with each other. And then there are a lot of studies in the model organisms. We can also, through the ortholog relationship, we can also map those to human and then, for example, guess the interaction relationship in human. And we can also do phylogenetic profiling. So meaning you have a lot of proteins in each organism, right? And then you look at the existence, whether this protein exists in organism A and B or etc., right? And then after you do this for a lot of hundreds of organisms, you will be able to see some proteins tend to co-occur together. For example, in this map, in this table, can you tell me which two proteins are more likely to be interact with each other than the other proteins pairs? Is it B and C?
B and C in organism.
Yeah, exactly. A and C because always appear together, right? So if you need two proteins to interact, they have to coexist in that organism in order to interact. And similarly, through the gene expression or protein expression experiments, if we see two proteins always come up together, and then we can infer the interaction relationship. But of course, those are all computational approaches just help us to make inference needs to be validated. Yeah, and another way is, you don't want to do experiments and you don't know how to do the computational inference, but there are plenty of protein-protein interaction databases that you can use to download those information. And here I list quite a few databases that you can, I don't want to go through them individually, but it's in the handout, and then you can get those, get to know those resources by yourself after the class. And for the protein-DNA interaction, so basically we want to establish the relationship between the transcription factors and the target genes. And the experimental approaches include the chip-on-chip, which is the early version of the study. Now people are usually doing ChIP-seq. And the computational approach, and we can do some promoter sequence analysis through the motif analysis, or we can do reverse engineering from MRI profiling data. And also there are databases that we can get this type of information from the Transfect. I think this is now commercialized, but the Jasper is still an open source resource that you can use. And the metabolic networks, those are the networks that have been very well studied for a long time and very well established database for this type of networks. And the two commonly used ones include the KEGG and the Metaseq. These are two well used metabolic network or pathway databases. And for the co-expression network, which is also typically used, I mean, this is mostly from the computational analysis. You start with the gene expression or protein expression matrix. Each row is a gene, each column is a sample. And then you can use one of these methods to do co-expression network inference. For example, the WGCNA, what you do is you calculate for each pair of genes, you calculate the co-expression relationship, and then, for example, through the Pearson coordination, and then you get a score, and you get a weighted network. And then what they did was to raise the coordination to a certain power to further discriminate the highly correlated ones from the lowly correlated ones. And the NetSam package we developed, so basically try to convert this weighted network into some unweighted networks, because there are certain graph algorithms that we can use, can apply to the unweighted network, but cannot easily apply to the weighted network. In this case, we can think about a k-nearest neighbor approach. So basically for each of the nodes in the network, we can ask what are the, let's say we are talking about a two nearest neighbor network, and we are asking in this network, what are my two best friends? And then we get, for each gene, we vote for the two best friends for each gene. And then when you think someone is your best friend, the other guy may not think the same, right? And then we also remove those relationships. We only keep the ones that are mutual. You think I'm your best two friends, and you also think so. So basically, this can give you from this network to a very robust relationship with but it's unweighted network now. So Arachne is also another very popular tool to use to build a co-expression network, rather than using Pearson coordination or Spearman coordination, deriving the relationship between two genes based on mutual information. So that is also a good idea, because the mutual information can capture different types of relationships, not only the linear relationship or monotonic relationship. There are more types of relationships that can be captured by the mutual information. So let's say you go through all this, and then you were able to build a network, right? This was what happened maybe in early 2000. There are a lot of experiments that have been done, and the people start to build the protein-protein interaction networks from the early time for the yeast, and then also the human protein-protein interaction networks through experiments have been published. At the very beginning, people get very excited. Oh, this looks great, and we get a lot of information. But then, if you look at this, people start to realize these are just hairballs, right? It's beautiful to look in a way, but what can we gain from this? So then the next question is, if you have the network, what can you learn from the network? And in order to do that, maybe I will introduce a few more terms in order to better understand these networks. The first is the degree. The degree means the number of links or edges each node has. For example, we can look at this node, this purple node in this network, and basically it has 3 links, and it is degree of 3. This is for undirected network. But if it's within a directed network, each node have incoming degrees and also outcoming degrees. And for example, this MET-R, this gene, it has out degree of 1 to 3, but in degree is 1. So degree is a very simple but important measurement for the nodes or genes in the network. Because if you look at this, you would think this is kind of in the more center position of the network if it has higher degree, right? So it's a very simple indication of the centrality of each node in the network. And the second thing we want to talk about is a path. So that's how we start to explore the relationship between two nodes in the network, right? For any pair of nodes you pick, and you can find paths that link these two nodes. For example, here, and we can find this path, which include two edges, and then we can also go 1, 2, 3 here, or we go 1, 2, 3, 4, 5 here, right? So that a lot of different paths that you can find in your network. And the one with the shortest total length of a path, for example, from this node to this node, I think this path with two links is the shortest path. This is called the short path between them. So with the understanding of the degree and the path, then we can start to explore some of the property of these networks. I think after we get a network, the first thing we want to do is to understand how the network is organized, what are the characteristics we can learn from those networks. The first one, the first property we want to talk about. So maybe I will show you this example. I mean, these are two networks. Each of them have 130 nodes and 215 edges. So the number of nodes and edges are the same. But if you look at these two networks, they're not they're actually quite different, right? So one thing is that this network is more homogeneous, meaning every node is very similar to each other. So basically, they have the same number of connections. The 5 red nodes basically with the highest number of links only reach 27% of the other nodes. But if you look at here, the nodes are very heterogeneous, meaning some of them have a lot of links, but most of them only have 1 or 2 links. So in this network, the 5 red nodes with the highest links, they can reach 60% of the other nodes in the network. So this is, that means the nodes are very similar in this network, and the nodes are not similar in this network. And in reality, most of the real-life networks, like social networks or even biological networks, they usually have this organization rather than this one. And Prabha, who is the kind of very important person in network analysis, he named this network scale-free network, and this is basically a random network. If you randomly connecting nodes, this is what you get. But the real-life network are not like this, they are more like this. This is called the scale-free network. So this can be probably best understood in social networks, and we can immediately understand what are the hubs in the network, right? Of course, those are the people that are celebrities, like the stars, and for example, if you look at the social network, they of course have a lot of connections, and for us guys, I mean, maybe we don't know too many people, and we only have a few connections. So the hubs are the celebrities in the social network. But biological networks are also have this scale-free organization. So of course, and people interested to know, I mean, what are the hubs in the biological networks? Do they play a different role in the biological processes than the other nodes in the network? So in early 2001, there was an interesting study. So at that time, there were a genetic study in yeast, basically try to delete each individual protein in the yeast proteome and try to see the impact of those proteins, deletion of those proteins. And they found some of these red proteins had a lethal impact, meaning if you delete that protein, the cell will die. And some of them, does not cause much impact, or some of them only cause slow growth. But at that year, I think the protein interaction network of the yeast have also been published. So this group tried to combine these two types of data and see, and now I group the nodes in the network based on the number of links or edges or degree they have in the network. And here is nodes with only 1 link, and here is a node with 20 links. So basically, you group them based on the number of links. And then they look at the percentage of the nodes in that category that has a lethal impact after when the protein is deleted. And you can see a very nice positive correlation between the number of links of a node and the percentage of the lethal proteins in that category. That means the hub proteins, meaning the nodes that have more connections in the network, when it's deleted, it will have a stronger impact to the cell itself. And after that publication, and people keep exploring what are the other possible properties of the hubs in the biological networks. And in this review article by Mark Waddell in 2011, and he summarized the major findings. So basically, the first study showed the hubs correspond to the essential genes, and they tend to be older proteins, and usually they evolve more slowly than other proteins in the network. They have tendency to be more abundant, and they have a larger diversity of phenotypic outcome when it's deleted. They could have different types of functional, that means they may be involved in more different types of functions. So, and then you may wonder why the cells choose to establish, or the evolution has evolved into this scale-free network rather than a random network, right? Does this give the cell any benefits? And indeed, if we think about a network like this, and if it's a scale-free network, let's say the mutations, we know that the mutations or random attacks on genes, right? But if the mutations occur randomly across the genome, it could hit any of the proteins. But most of the proteins are the proteins only with 1 or 2 links. So mutation in these proteins will not affect the cell as a system, right? So it provides the cell the robustness to survive. So meaning mutations typically don't have an important consequence because it does not make significant impact to the network as a system. But this also gives us some, we can also think it this way. Now, if we want to try to kill bacteria in the cell, and what should we do? And in that way, we can think about the targeted attack of the central node, meaning the hubs, for example, in the bacterial network, because if we attack these important nodes, then it will destroy the bacteria. I mean, that's one way we can think about how to prioritize genes when we want to treat a disease. We may try to kill fungi or kill bacteria in human. That's the first property of the network, is the existence of hubs, or the scale-free property of the network. The next thing I want to talk about is the small world network. This was originally studied in the social network context by a scientist called Stanley Milgram. I think he was a social scientist in Harvard, and he did this experiment in 1967. So the idea is he wants to understand how people in the are connected, right, to each other. Of course, now it's easy because we have the internet, and since we know how to do this. But at that time, I mean, it's actually very difficult to do this. How can you even think about, a way to estimate or to understand how people are connected to each other? I think he came up with a very interesting experimental design. So he prepared 160 packages, and he gave those packages to random people in a very small city, Omaha, Nebraska, in the US. And then he gave these packages to random person in this small city, and they asked them to try to send the package to a stockbroker in Boston, which is far away from Omaha, right? And then you cannot directly send this to him because you don't know him. You have to, every time you have to pass this to somebody you know. This is his instruction. So, and then at the end, he collected all the letters from this stockbroker, and then he counted how many times is required for the letter to reach the stockbroker from the original place. And surprisingly, the average number of passes to reach him was 6. So that is the famous 6 degree of separation. I think probably some of you have heard about of this phrase came from his study. And then through this study, he understands, well, although it looks like everyone is so far away from each other, especially when you think about it, it was in 1967, right? I mean, maybe nobody knows each other, but now you see, for any person, you don't really have a good connection, and still you can reach him in 6 steps. So this is what he calls the small world network. And if we look at the biological networks, it's also in the average path length between any 2 nodes, and it's between like 3 to 4. Actually, it's even smaller than the social network he estimated at that time. I think one reason with biological networks use a small network structure is probably because it can pass information more efficiently. And the next property I want to talk about is the motifs. So when we talk about motifs, it's patterns that keep occurring in a system more often than random chance, right? So if we think about 3 nodes in, what are the possible relationship between them? There are actually 13 different types of relationship between 3 nodes if they are somehow connected. And then, for example, this pattern is very well studied. It's like the feed-forward loop, right? Node A can have a positive relationship with this node B, and then it also through node C, it also has A, C, B, right? So there is it's called the feed-forward loop, and this is called a feedback loop. So basically, it goes around. And then for each of these, maybe let's say this is a real network, and then you can count all the how many times you see a feed-forward loop, the 3-node motif in this small network, and then you want to know whether this is a motif. Because the definition is statistically more frequent than random chance, right? What you can do is to randomly shuffle the nodes in this network and build some random networks. And then you can, after you do the shuffling, you can count the number of the feed-forward motifs again. And then, for example, here we see a lot more feed-forward motifs in this network than the random networks. And there is a study in E. coli, transcriptional regulatory network, because in engineering, we know that the feed-forward loop and the feedback loop both are very commonly used, to regulate a system, right? But in the biological network, like the transcriptional network, they actually found that the feed-forward loop, they observed 42 in the E. coli network, but they only didn't observe any feedback loop in that network. And this is significantly more than what you would expect by chance. So that means this is kind of a motif or the way of organizing the nodes that the biological system actually use, but not the, the feedback loop. And the next one is the modularity. So this basically says the genes and or proteins in the network tend to form groups rather than randomly connected to each other. So in the transcriptional networks, these are, of course, the transcriptional modules, meaning the target genes, of course, if they are targeted by the same set of transcription factors, they form a group, right? And in the protein interaction network, like this one, we can see there are groups of proteins in the network, and these are usually the protein complexes in the network. And the signaling networks, we can have the signaling pathways that represent the modules in those networks. And also, the modules are not only occurred separately, they're also organized in a hierarchical way, meaning there are small modules that get connected to each other to form a relatively larger modules, and then eventually reach the whole network. And we can think about the protein complexes, and the two complexes may interact with each other to form a relatively larger network, and eventually we know the 6-degree separation, right? Everything is actually connected. So those are the 5 different properties associated with network that have been revealed through a lot of studies.
In today's lecture, you are introduced to various types of protein-protein, protein-DNA and other biomolecular network analysis. These interactions can be either experimentally determined or through the use of computational software predictions. Databases like DIP, MINT, BioGrid, they contain information of various protein-protein interactions. Metabolic networks can be studied using databases like KEGG or MetaSic, while co-expression networks can be computationally determined. Pearson correlation coefficient is a direct indicator of lethality and connectivity. It is important to note that biological networks generally follow the characteristics of small world networks. I hope this information is giving you more clues and ideas how you can utilize these available resources and do more protein-protein and protein biomolecular interaction and network analysis for your own data set. In the next lecture, we will learn more about the visualization of network, and Dr. Bing Zhang will continue his lecture and show you how to use various tools to do data visualization for network analysis. Thank you.